persistent effects of the anesthetics is we've turned the brain off profoundly. We've left it there. Arguably, these are pathologic states. They're not natural states. Maybe we should just flip the switch and bring it back on. All right, so Ken's worked on that a fair amount. And I'm just going to illustrate one particular case where he's done this. So again, this is one of our willing rat volunteers. And so he's stretched out there, breathing isoflurane. And he has an IV in his tail vein. And so he's been there for about 20 minutes or so. And Ken's going to give him just a saline injection, basically to show you that if you inject saline, nothing happens. All right, so let a little time go by. Let a little time go by, and now Ken's going to inject, well, he lists it as a test drug, but I'll just tell you it's methylphenidate, it's Ritalin. All right? So now just to flush it into the animal, and then you see the response. Okay? So those of you who do these sorts of experiments far more than I do know that this is the writing reflection that you wanted to turn over for it to really count. But remember, he's still breathing anesthetic in, and he's receiving, and he's received something which has certainly changed his behavior, let's say. Now he's got to turn over. He's sort of caught in the things there. He does eventually make it. I've seen one of the video. Okay, he made it. All right, good. All right. So why would you do that? Why would that make any sense? Okay. So here's the logic. All right. Let me just remind you of a pathway here. So the ventral tegmental area is one of the main sources of dopamine in the brain. All right. It projects the hippocampus, the amygdala, the frontal cortex. All right, now this pathway is often thought about mostly for reward. People do reward studies up and down the wazoo, looking at amygdala activity and sort of VTA activity in cases where they're studying reward. But it's also a very important pathway for cognition. All right. Now there are two other sort of dopamine pathways here. The one from the substantia nigra is the one which is associated with Parkinson's disease. And actually, in patients that have Parkinson's disease, they actually have a diminished amount of dopamine being produced in the ventral tegmental area as well. And there's also the ventral periaqueductal gray, all right? Now, so what is Ken doing? So he's giving Ritalin. And what Ritalin does is block the reuptake of dopamine and probably norepinephrine also, maybe histamine, but let's say mostly dopamine, okay? And so what you do is it's basically like giving an arousal signal. It's like taking advantage of this pathway. All right, but what's the physiology of this like? And he's also done studies, in fact, where he's used electrical stimulation to, induce this arousal from anesthesia. All right, but let's just go back to this diagram. So this is somebody awake. All right, so look at this guy right here. This is a neuron in the cortex. He's got all these people telling him what to do. He's got an interneuron here, inhibition. He's got all these guys coming up from the brainstem, and being awake is a balance between inhibition and excitation. Okay? Now, when you anesthetize with propofol, or let's say the isoflurane, you shift to mostly inhibition. Okay? And you're anesthetized. All right, now, if you take this anesthetized state, right, and now you add Ritalin to it, you're not blocking the effect of the propofol. What you're doing is you're increasing the excitation, all right? So what then happens is the anesthetized state goes to this awake state. All right, so we're not blocking the effects of the propofol, we're just driving an arousal pathway, all right? So Ken has just received permission to, to study this in humans, from the FDA, all right? So just some concluding thoughts. So a major theme of what I've been suggesting is I think we as anesthesiologists have to go back and, get our neuroscience down and our, not only sort of the circuits and some neurophysiology, but also the clinical neuro exam. I mean, I do a very quick mini neuro exam on pretty much all of my patients, particularly when they're recovering. What you can learn is just amazing. We can talk about that maybe during the break. I think the EEG is really the brain's electrocardiogram. There's a lot of information in there. And remember, I'll tell you something. The signal-to-noise ratio of the anesthetized brain is far higher than anything like you'll see in sleep. I mean, it's amazing. And so, again, the fact that those large changes couldn't give us insight into how the drugs are working, it would be very surprising. Let me just say it that way. And I think that, as I've sort of suggested, if we combine these sorts of neurophys insights with the pharmacology, I think that's kind of the next frontier, particularly for those of you who are interested in drug pharmacology. So I'll stop there. Thanks.
We do have time for a couple of questions. See your first hand up there.
Mark.
Emery, early versions of some of our brain function monitors had posterior electrodes, and they abandoned them because the anesthesiologist didn't like them, because they couldn't make them stick. But if we went back to looking at the back of the head, wouldn't that improve our ability to monitor the brain under anesthesia?
It could. It certainly could. I mean, I think there's information there. There clearly is information in the dynamics. And I think it's just, it's obviously something that, you and I don't control, as the people who are in the operating room. It's something that the companies control. But from a purely information standpoint, yes, there would certainly be more information in doing that. Good question.
I think it's a very good talk.
Thank you.
Actually, the paper you published last year in PNAS actually talk about the disconnection between the different areas and surface by using the electrode in patients, three patients, right? It's a very good research work.
Thank you.
You mentioned actually the fragmentation has occurred during this process, propofol infusion, to induce unconsciousness during anesthesia. If so, I think it means that the disconnection has been occurred among the different areas in the cortex, all the different nucleus in the brain. If so, that means the network of the neurological relationship between many nucleus has been disconnected. If so, I think it's something new for us, because we think about if a projection has been established between the different nucleus, such as hypothalamus nucleus and the cortex. That means if we stop one pathway, it should be occurred in many different areas
Right.
at the same time.
Right.
So how to explain it?
Well, so the issue is like what's really going on. And let me tell you, to be honest with what I've shown here, and maybe even the stuff that we published, is really on the tip of the iceberg. Because we have other patients where we have electrodes in a number of areas. Like we have patients that have like these electrodes in like hippocampus. And what's probably happening is something like the following. These are some of the larger dynamics that you see. But if you look in areas like the hippocampus or like the medial temporal lobe, you'll see like local oscillations, right, that have nothing to do like with these guys up here, like these big oscillations. So if you think about it, you have the brain is this huge network, and it's got all these interconnected loops. So if you just look at it from a dynamical system standpoint, if you try to push that system, you can probably make it break into many sorts of different types of oscillations, and I think that's basically what we're seeing. Now, and just to be clear about what we were saying with the slow oscillations, that at the moment is intracortical, all right? Now, but I will tell you that if you look at the slow oscillations in sleep compared to the slow oscillations under propofol anesthesia, the ones in sleep are coordinated. And they'd have to be, right? Otherwise you couldn't dream and this sort of thing, right? But the ones under propofol, they're not coordinated. They're actually quite uncorrelated.
So Emery, the problem of quantitative EEG analysis and trying to infer what's going on in space over the cortex to explain the EEG phenomenon is a relatively
Mhm.
So where do you see this going? What kind of information can we get out?
So, I mean, I honestly believe there's a huge amount of information we're not using. So just doing two things. But think about how the process has worked up until now, right? The EEG has been taken and then turned into an index, okay? And so then we're trained to look at the index. And the idea is, we see some exceptions that bother us, maybe ketamine, maybe nitrous, maybe we can't quite use it on kids, this type of thing, all right? So put that away. Go back and look at the raw EEG. Go back and look at the spectrogram, all right? Quite simply. There's a huge amount of information there. And I didn't show it here, but every drug has a different signature. It changes depending on how you administer it, changes probably depending on how old you are, depending on how sick you are. They're very, very robust covariates. So once we add that sort of thinking to our practice, I think it opens up a wellspring of possibilities. And I think that, and just to be fair, remember, if you look at quantitative EEG, right, the neurologists don't do quantitative EEG. They don't look at the spectrogram. You'll ton through, chart after chart. Here's this pattern, here's that pattern, right? But if you, both the patterns and the spectrogram, huge opportunity.
Going to try two other quick questions before we break.
About burst suppression, do you think burst suppression is a mandatory condition compatible with adequate anesthesia? Because my impression is that once you have 20 or 20% of burst suppression and you maintain a constant concentration of anesthetic drug, that the tendency is that this burst suppression increases, that there's a wind-up phenomenon. And as I now learned that there might be a relation with energy, ATP or energy
Right.
provided, isn't that potentially harmful for, I don't know, elderly that are at risk for? So I'm a bit concerned whether burst suppression is not a too deep sign of EEG change for titrating anesthesia.
No, it's a great question. I mean, so the first answer is I don't know. I honestly don't know whether it's too deep or too light, okay? But just the factoids, okay? So the first factoid is, it is used to treat medical coma, okay? So if we're going to use it there, why don't we do it in a principled way and control it, okay?
The problem is with the coma is generally that these are damaged brains.
No,
Want to protect the
I'm with you.
now these are healthy. Our coma patients have healthy brains.
I get it. I understand. No issue with that. Whether or not it's too deep or whether or not it's too light, I think is an open question. You could argue it both ways, right? If it's creating some sort of metabolic reserve, right, maybe we should send everybody there, if that's the case, right? Okay? Or if we can clearly anesthetize you and give you adequate anesthesia at a lighter plane, maybe we should do that. Either way, the induction of these states is not natural. So I think it's an open question to figure out, but the only way we can figure that out is if we get behind the neuroscience of, essentially like what's, and I honestly do not know. But I can tell you this, the older patients reliably go right into burst suppression, and they will just stay there. And so one of the things, and I've been able to just reduce the dosing of my drugs tremendously, cutting it to like a fourth of what I might otherwise give based on sort of standard profiles, just because it's very clear from being unconscious. Now, movement's a whole other thing, but in terms of being unconscious, right, you have more than enough there with the current regimens, because the patients will be in burst suppression.
Good. Take one last thing. Sorry, John's been waiting patiently.
About 10 states at the end of that class.
Mm-hmm.
You were giving lethal candidate, weren't you?
Right.
You were giving it intravenously?
Right.
How does it cross the blood-brain barrier to produce the effect when you charge one?
I don't know. Happy to say I don't know. That's my homework assignment.
With that, we're already invading our brief break. I want everyone to get a little caffeine. Just get us through that.